We've just navigated the complex stages of development and seen how childhood experiences can cast long shadows into adulthood. We know the grim statistics. Abuse begets abuse. Witnessing violence correlates with PSD. Losing a parent increases depression risk. It's a pretty bleak picture, doesn't it? But hold on, reality isn't quite that simple. Look around you. Countless individuals who faced unimaginable hardships in their youth are now leading relatively stable, even fulfilling lives. The question hanging in the air, the one that keeps researchers and clinicians up at night, is this: what makes some people resilient? How do they bounce back from the blows life deals them? That's the puzzle we're tackling today. Now, before we get lost in the weeds of individual trauma types, let's zoom out a bit. Think about it like this. If you were tallying up the hits a child takes, does it matter more whether they got hit with a baseball bat or a feather duster, or how many times they were hit? Often, the sheer number of adversities is a stronger predictor than the specific type. For instance, being both sexually abused and forced to witness violence leaves a deeper scar on adult prospects than experiencing either one alone. It's a cumulative effect. And it's not just about the negative stuff. Protective factors are crucial. A child growing up poor might fare better if their family is stable and loving, compared to one in a broken, acrimonious home. The basic logic is pretty stark. The more different kinds of adversity pile up, the dimmer the odds look for a bright, functional future. It's like stacking risks, each one adds weight. Okay, let's talk about the extreme end of the spectrum. Imagine a scenario where almost everything goes wrong. No consistent mother figure, minimal peer interaction, sensory deprivation, cognitive neglect, and maybe even malnutrition. Sounds like science fiction, but it was the reality for thousands of children in Romania during the 1980s. Under Nicolae Ceausescu's bizarre policies banning birth control and abortion while demanding large families, institutions overflowed. Kids were warehoused, neglected, forgotten. When the regime fell in 1989, the world learned what was happening. Many kids were adopted out, thankfully, and studies began, most notably by Charles Nelson at Harvard. These kids became, unfortunately, poster children for just how devastating childhood can be when it's stripped bare. This isn't just a historical footnote, it's a stark natural experiment on the effects of profound deprivation. So, what happened to these Romanian orphans as they grew up? Well, largely, the outcomes fit the grim expectations. We're talking about significant challenges: lower IQ scores, difficulties with cognitive skills, and serious problems forming attachments, sometimes described as bordering on autistic traits. Anxiety and depression were rampant. And here's a critical finding: the longer a child remained institutionalized, the worse their adult prognosis tended to be. It wasn't just about having been there, it was about how long. This provides powerful evidence for the importance of early, consistent, nurturing care. Deprivation, especially prolonged deprivation, doesn't just cause temporary setbacks, it can have lasting, measurable impacts on mental health and functioning. But the story doesn't end with behavior. It goes deep into the hardware. What did these studies find when they looked at the brains of these individuals? The results were quite striking. On average, these adults showed decreased total brain size, less gray matter, less white matter, reduced metabolic activity in the frontal cortex, impaired connections between different brain regions, and even smaller sizes of specific brain areas. It's a litany of deficits. Now there's one interesting exception. The amygdala, the brain's fear center, was actually enlarged. Why this specific pattern? While the exact reason for the amygdala enlargement isn't fully understood, the overall picture is compelling. It strongly suggests that severe neglect and deprivation during critical developmental periods can physically alter brain structure and function, potentially explaining the observed behavioral and emotional difficulties. It's a biological echo of an early life trauma. All right, let's shift gears slightly from the extremes of deprivation to the broader canvas of culture. As we'll explore more deeply in Chapter 9, culture profoundly shapes our behavior, both good and bad. Two fundamental points to grasp right now. First, childhood is the prime time for cultural transmission. It's when we learn the rules, norms, and values of our society. Second, parents are the primary agents, the gatekeepers, mediating this process. They're the first teachers. And just to emphasize how diverse this process is, consider the sheer variability across cultures in basic child-rearing practices. Think about things like breastfeeding duration, how much physical contact infants have with caregivers, how often they're spoken to, how quickly their cries are responded to, or even when they start sleeping alone. There's no single right way written in the stars. Now, whenever we look at cross-cultural differences, it often brings out the competitive spirit in us, doesn't it? Parents start wondering, do other cultures really do a better job? Is there some magical recipe out there? You know, the perfect mix of the Kwakito baby diet, the Trobriand sleeping program, and the Ituri approach to educational videos. We humans love searching for that anthropological ideal of child rearing. But spoiler alert, there isn't one. As the anthropologist Meredith Small points out, cultures, starting with parents, raise children to become adults who behave in ways that are valued by that specific culture. It's not about finding the best way objectively, but about understanding the logic within each cultural context. Different cultures aim to produce different kinds of functional adults. So, how does this cultural shaping begin in earnest? One major avenue is parenting style. Interestingly, the most influential framework for understanding parenting styles didn't initially spring from observing parents directly, but rather from analyzing broader cultural patterns. Think back to the post-World War II era, trying to understand the roots of fascism. Scholars like Hannah Arendt and Theodor Adorno were grappling with why figures like Hitler and Mussolini emerged. Adorno, in particular, explored the personality traits associated with authoritarianism, things like extreme conformity, submission to authority, aggression, and hostility towards independent thought. He argued these often had deep childhood roots. This line of thinking heavily influenced Diana Baumrind, a psychologist at Berkeley, who, in the 1960s, started classifying common parenting approaches based on these ideas. Baumrind identified three main parenting styles, which have since been replicated and adapted across cultures. First, there's authoritative parenting. Think clear, consistent rules, but explained logically. Because I said so is frowned upon. There's flexibility, a focus on praise over punishment, and kids are encouraged to voice their opinions. The goal is fostering potential and autonomy. The typical outcome? Adults who are generally happy, emotionally mature, fulfilled, independent, and self-reliant. Then there's the opposite end, authoritarian parenting. Here rules are numerous, rigid, and often arbitrary, enforced primarily through punishment. Emotional needs, low priority. The rationale is often preparing kids for a harsh world. The result? Adults might be narrowly successful, obedient, and conformist, but often harbor resentment, aren't particularly happy, and struggle socially because they didn't learn much through experience. Finally, permissive parenting. Few demands, rules are rarely enforced, and the child basically sets the agenda. The adult outcome? Often self-indulgent individuals with poor impulse control, low frustration tolerance, and again poor social skills due to a consequence-free childhood. Boomers supposedly invented the 60s thanks to this one. Baumrind's original trio was later expanded by Stanford psychologists Eleanor Maccoby and John Martin to include a fourth style, neglectful parenting. This style is characterized by low demands on the child and low responsiveness from the parent. Think uninvolved, detached. This gives us a neat 2x2 matrix. Parenting can be high or low in demands and high or low in responsiveness. So you get the four quadrants: authoritative, high demand, high response; authoritarian, high demand, low response; permissive, low demand, high response; and neglectful, low demand, low response. It's a useful framework. Importantly, research suggests that each of these styles tends to produce adults who adopt a similar approach in their own lives. Of course, different cultures might value these different styles more or less, but the underlying patterns seem to hold. Now, let's introduce a major challenge to the traditional view of parenting's role. Enter Judith Rich Harris and her controversial book, The Nurture Assumption. Harris, interestingly, wasn't an academic psychologist with a doctorate, yet she sent shockwaves through the field. Her central argument, the importance of parenting in shaping a child's adult personality, is massively overstated. Instead, she argued that once kids pass a surprisingly young age, their peers become the dominant influence. She offered several arguments. Parental influence is often indirect, mediated through peers, like how neighborhood quality affects kids via peer groups. Peers influence language development, think accents. And importantly, she pointed out that in other young primates, peer socialization is key. The book caused a huge stir, partly because headlines distorted it. Psychologist says parents don't matter. While the dust has settled, the consensus now leans towards acknowledging that peer influence is probably underappreciated, but not dismissing parenting entirely. Parents still play a huge role, including by influencing which peer groups their kids end up in. So if peers are so influential, why? What makes them such powerful teachers? Well, peer interactions are essentially boot camps for social competence. They teach kids how to navigate complex social situations, figure out who's a friend and who's a foe, and understand where they fit into the social hierarchy. And the primary tool for this learning, play. Seriously, play is the greatest teaching tool ever evolved for acquiring social skills. Think about it on different levels. Writ large, play encompasses a whole range of behaviors that train individuals in social dynamics. Writ medium, it's like running simulations, fragments of real-life interactions, allowing kids to safely try out different roles, practice social scripts, and improve motor skills. Writ small, endocrinologically, moderate stress during play is actually beneficial. It's a form of stimulation. And neurobiologically, play seems to be involved in pruning excess synapses, refining neural circuits. It's a multi-layered learning machine disguised as fun. This idea of play as fundamental to human development isn't new. The historian Johann Huizinga famously characterized humans as Homo ludens, man the player, highlighting our structured, rule-bound games. And play isn't just a human thing, it's universal among socially complex species. It peaks around puberty and involves remarkably similar behaviors across different animals, like dominant dogs, crouch to signal playfulness, which translates to other species like baboons, presenting their rear. Why do animals engage in play? Because it's vital. Think about it. Playing means foregoing foraging, burning calories, making themselves conspicuous to predators. Yet young organisms persistently squander energy on play, even during famines. Why? Because it's intrinsically pleasurable. Dopamine pathways light up during play. As psychiatrist Stuart Brown puts it, the opposite of play isn't work, it's depression. The real challenge is figuring out how the brain encodes the reinforcing properties of such a diverse range of activities, from intellectual sparring to silly noises. One particularly notable type of play involves rough and tumble activities, wrestling, headbutting, play biting. Harlow called it that. We typically see males engaging in this more frequently than females, and interestingly, exposure to higher levels of prenatal testosterone seems to boost this behavior. So, what's the purpose? Is it just practice for the status tournaments of adulthood? Or are they already in the arena, learning social dynamics through simulated conflict? The likely answer, as always, is probably a bit of both. It's a complex blend of learning social hierarchy, practicing physical coordination, and perhaps even rehearsing responses to threat and aggression in a safe context. It's not just random chaos, it's structured learning disguised as play fighting. Beyond immediate peers, the neighborhood itself acts as a silent teacher, constantly communicating cultural values. Think about the cues kids absorb daily. Is there trash everywhere? Are houses falling apart? What's most common, bars or churches, libraries or gun shops? Are the local parks safe places to play? What messages do billboards, ads, and bumper stickers promote? Religious salvation or materialism, kindness or inclusivity? Kids aren't just passive recipients, they're constantly interpreting these environmental signals. It's a form of implicit learning, absorbing the prevailing ethos and norms of their community without anyone explicitly teaching them. The neighborhood becomes a powerful, albeit subtle, agent in shaping behavior and beliefs. Let's zoom out again to look at broad cultural differences, like the widely studied contrast between collectivist cultures, often East Asian, and individualist cultures like the US. Collectivists emphasize interdependence, harmony, fitting in, and group needs. Individualists value independence, competition, and individual rights. These differences permeate parenting. Studies show mothers in individualist cultures tend to speak more loudly and animatedly, see themselves as teachers, dislike bored children, and encourage assertiveness and autonomy. Show them a cartoon of a school of fish with one leading, and they'll praise the leader. Mothers in collectivist cultures, conversely, spend more time soothing and comforting, value calmness, and games focus on cooperation and sharing. Show them the same fish cartoon, and they'll likely say the leader did something wrong and is now excluded. This difference logically impacts when kids develop theory of mind, the ability to understand others' perspectives. For collectivists, taking others' viewpoints is paramount. Interestingly, Japanese kids play more violent video games but are less aggressive, possibly because they play near parents, not alone, and games often have prosocial themes. Culture shapes even media consumption. Another fascinating cultural dimension is the concept of cultures of honor. These cultures strongly emphasize rules of civility and hospitality, but they also carry an expectation of retribution for perceived insults to one's honor, self, family, or clan. Failing to respond aggressively is considered shameful. Think vendettas, revenge, and tragically, honor killings. Cheeks aren't turned in these cultures. The American South is often cited as a classic example, but these exist globally. They often correlate with historically contested environments or perceptions of victimization. Parenting in these cultures tends to be authoritarian, and children raised in them are often more aggressive, especially after experiencing an honor violation, and strongly endorse aggressive responses to such scenarios. It's a cultural system built on vigilance and readiness for retaliation. Finally, let's consider class differences within Western societies. Unlike a baboon cub learning its rank solely from its mother, human lessons about status are far more nuanced. We use implicit cues, subtle language, invoking history. My grandparents couldn't even, and expressing hope for the future. When you grow up, you'll be able to. Human parents teach kids not just what to do, but what to dream about. Research shows class differences in Western parenting often align with Baumrinn's types. Higher SES parents often lean towards authoritative or permissive styles, focusing on exploration and facilitation. Lower SES parents often lean towards authoritarian styles, reflecting themes of protection against constant threats and preparing kids for a tough world. Adri Kusaro's classic study in Queens, NY, beautifully illustrates this. In poor neighborhoods, parenting was defensive, aiming to shelter kids, using toughening tactics like teasing. In working-class areas it was offensive, aiming for upward mobility, using metaphors of progress. Upper-middle-class parenting focused on psychological fulfillment and individuation, using gentle requests and emphasizing potential. Each class had its own distinct cultural logic for raising kids. Okay, we've talked about environments after birth, but let's rewind significantly. Environment doesn't begin at birth, it starts much earlier. Consider the womb itself as an environment. And guess what? Fetuses are soaking it all in. Public imagination was captured by charming studies showing fetuses hear outside sounds, taste amniotic fluid, and remember these preferences after birth. Inject lemon saline into a pregnant rat's amniotic sac, and her pups prefer that flavor. Spices consumed by pregnant women can flavor the amniotic fluid, potentially influencing food preferences later. Anthony DeCasper's work showed newborns recognize and prefer their mother's voice, learned in utero. Even elements of language are picked up. Newborn cries mimic the rhythmic contours of their mother's native tongue. Near-term fetuses can even distinguish between nonsense syllables like Biba and Bobby. One study even showed newborns whose mothers read The Cat in the Hat repeatedly in the third trimester preferred that book's rhythm over a similar one. Now, while these findings are fascinating, they're not the core of how major behavioral influences operate. But they do show prenatal learning is happening. Beyond just sounds and tastes, the fetus is bathed in hormones secreted by its own developing glands. This brings us to a crucial distinction: organizational versus activational effects of hormones. We touched on activational effects earlier, short-lived impacts, like a hormone surge affecting behavior for a day or two. But hormones in the fetus have organizational effects. These are profound, lasting changes that wire the brain for life, happening during critical developmental windows. Around 8 weeks post-conception, human fetal gonads start secreting steroid hormones, testosterone in males, estrogen and progesterone in females. Crucially, testosterone, along with another hormone called anti-Müllerian hormone, works together to masculinize the brain. This isn't just a temporary tweak, it's a deep structural programming event. Now brace yourselves, because the biochemistry gets wonderfully messy. First, in many rodents, the brain isn't fully differentiated at birth, so these hormonal organizational effects continue after birth. Second, here's a mind-bender. Surprisingly few of testosterone's effects in the brain actually come from binding to androgen receptors directly. Instead, testosterone enters cells and gets converted into estrogen. Yes, estrogen. This estrogen then binds to intracellular estrogen receptors, triggering the masculinizing changes. So testosterone achieves its masculinizing effect in the brain largely by becoming estrogen. Third, female fetuses are bathed in estrogen from both the mother and themselves. So, why doesn't this masculinize their brains? The likely culprit is alpha-fetoprotein, a substance made by the fetus that binds circulating estrogen and neutralizes it. Finally, the default state for a mammalian brain seems to be feminization. Unless testosterone and AMH are present, the brain naturally develops along female lines. It's a complex interplay of hormones, enzymes, and protective mechanisms. So given this hormonal setup, How much does it explain sex differences in behavior, particularly aggression? Robert Goy's pioneering work in the 1950s with guinea pigs provided compelling evidence. He treated pregnant females with testosterone. The resulting female offspring looked perfectly normal externally, but internally their brains were organized to respond to adult hormones in a male-typical way. When given a testosterone injection as adults, these females showed increased aggression and mounting behavior compared to controls. Estrogen, however, was less effective at eliciting female sexual behavior. This was a bombshell finding. Prenatal testosterone exposure organizes the brain to respond to adult hormones according to sex. This directly challenged the prevailing dogma that sexual identity and behavior were purely socially constructed, a view held by some sociologists and even parts of the medical establishment. It suggested that for intersex individuals, biological factors might be more decisive than previously thought. The guinea pig results were striking, but critics retorted, well, those are just guinea pigs. So Goy and colleagues moved to non-human primates, specifically rhesus monkeys, which show strong sex differences. Adult male rhesus monkeys, for instance, show a clear preference for playing with wheeled toys over stuffed animals, reflecting their generally higher activity levels. Building on hints that male monkeys are more active and engaged in rough and tumble play shortly after birth, long before puberty testosterone surges, Goy conducted a key experiment. Treating pregnant monkeys with testosterone produced daughters who looked female externally, but had male gonads internally pseudohermaphrodites. Compared to control females, these androgenized daughters exhibited significantly more rough and tumble play, aggression, and male-mounting behaviors. Importantly, not all behaviors were affected. For example, their interest in infants remained similar to controls. This demonstrated that prenatal testosterone does have organizational effects on specific behaviors in primates, but not a complete overhaul of the entire behavioral repertoire. Moving to humans, the disorder congenital adrenal hyperplasia offered a natural experiment. CA is caused by a mutation in adrenal enzymes, leading to excessive production of androgens, testosterone-like hormones, starting in fetal life. While the lack of other hormones causes metabolic issues requiring treatment, the excess androgens provide a unique window. Early studies by John Money in the 1950s claimed CAH girls showed extremely high levels of male-typical behaviors, low female-typical ones, and even elevated IQ, findings that were later discredited due to biased samples and outdated norms. However, contemporary research by Melissa Hines and others, comparing CAH girls who receive hormone treatment shortly after birth, normalizing postnatal exposure to controls, paints a different picture. These studies consistently show that CAH girls exhibit more rough and tumble play, fighting, physical aggression, prefer masculine toys, score lower on measures of tenderness, higher on aggressiveness, self-report more aggression, and show less interest in infants. Furthermore, CA women are more likely to identify as gay, bisexual, or transgender. Since the drug treatments normalize postnatal exposure, the behavioral differences strongly suggest that the prenatal androgen exposure itself caused organizational changes leading to these male-typical behavioral patterns. To further solidify the link between prenatal testosterone and male-typical behaviors, consider the inverse condition, androgen insensitivity syndrome, AIS. Individuals with AIS have XY chromosomes and testes that produce testosterone, but they have a mutation in the androgen receptor gene, rendering their bodies insensitive to testosterone. The testes churn out testosterone, but it can't get into the cells, or if it does, it can't trigger the usual responses. Consequently, the brain isn't masculinized by testosterone, and often the external genitalia appear female. These individuals are typically raised as girls. Puberty often reveals the discrepancy when menstruation fails to occur. Upon investigation, doctors discover the girl is genetically male. Most individuals with AIS continue to identify as female, though they are infertile. This provides powerful converging evidence. When human males fail to experience the organizational effects of prenatal testosterone due to receptor insensitivity, they tend to exhibit female-typical behaviors and identities. Together, CH and AAI strongly suggest a causal role for prenatal testosterone in organizing sex-typical behaviors in humans. However, a careful reader might raise some significant eyebrows at the conclusion that prenatal testosterone determines adult aggression. There are whopping big problems. First, remember the CH girls. They're not just prenatally androgenized, they're born with ambiguous genitalia, undergo multiple surgeries, live under intense medical scrutiny, and start hormone treatments immediately after birth. Can we really isolate the prenatal androgen effect from all these confounding factors? Probably not. Second, the AIS argument assumes the androgen receptor mutation blocks all pathways. But what about the estrogen pathway we discussed earlier? Shouldn't masculinization via estrogen still occur? Some monkey studies suggest testosterone effects don't always require conversion to estrogen. This means AIS individuals might still have some degree of brain masculinization despite the receptor issue. Compounding this, AIS individuals raised as females often show higher rates of non-heterosexual orientation and non-female gender identity, adding another layer of complexity. So, while the evidence points towards a role for prenatal testosterone, the picture is far from simple or deterministic. So, if the relationship is complex, how can we even measure someone's prenatal testosterone exposure? It turns out there's a rather quirky biological marker, the ratio of the second digit to the fourth digit, 2D4D. On average, men have a lower 2D4D ratio, meaning their ring finger is longer relative to their index finger than women do. This difference is measurable even in third trimester fetuses. Studies correlating this ratio with prenatal testosterone levels measured via amniocentesis show a clear link. Higher prenatal testosterone exposure leads to a more masculine ratio. Consistent with this, CAH females and females who shared a womb with a male twin, and thus received some of his testosterone, have more masculine ratios. Conversely, AIS males have more feminine ratios. This digit ratio difference exists even in other primates and rodents, and nobody knows precisely why it happens. It's just one of those fascinating biological quirks. There's even another marker related to background noise from the inner ear showing a similar sex difference linked to prenatal hormones. What does this 2D4D ratio predict about adult behavior? Well, it's not deterministic, but there are correlations. Men with more masculine ratios, lower 2D4D, tend to score higher on aggression and math tests, be more assertive, have higher rates of ADHD and autism, male bias conditions, and lower risk of depression and anxiety, female-skewed conditions. Their faces and handwriting are often judged as more masculine, and some studies link it to a decreased likelihood of being gay. Women with more feminine ratios, higher 2D4D, tend to have lower autism risk, higher anorexia risk, female-biased, be less likely left-handed, male-skewed, show less athletic ability, be more attracted to masculine faces, and be more likely straight or, if lesbian, to adopt stereotypical female roles. What's the takeaway? This provides some of the strongest evidence yet that A. Fetal androgen exposure has organizational effects on adult behavior in humans, just like in other species, and B. Individual differences in that exposure predict individual differences in behavior. The text quips, prenatal endocrine environment is destiny. But crucially, it adds, well, not exactly. These effects are small and variable. So far, we've focused on hormones secreted by the fetus itself. But the environment influencing the developing brain extends beyond that. The outside world constantly alters the pregnant woman's physiology, and these changes ripple down to the fetus. The most obvious example is nutrition. What the mother eats directly affects what nutrients the fetus receives. Extreme maternal malnutrition broadly impairs fetal brain development. Pathogens acquired by the mother can also cross the placenta. For instance, the parasite Toxoplasma gondii, often contracted from cat feces, can infect the fetus and potentially cause serious neurological damage. Then there's the well-known impact of maternal substance abuse, leading to conditions like fetal alcohol syndrome, and critically, maternal stress. Stress impacts the fetus both indirectly, leading to poorer diet, substance use, and directly, altering maternal blood pressure, immune function. Most importantly, stressed mothers secrete higher levels of glucocorticoids, stress hormones, which cross the placenta and enter the fetal circulation. This prenatal exposure to excess stress hormones has significant consequences. How do these excess glucocorticoids affect the fetus? Primarily through organizational effects on brain construction. Too much prenatal glucocorticoid exposure leads to decreased levels of growth factors, fewer neurons, and fewer synapses in the developing brain. Think of it like this. Just as excessive prenatal testosterone exposure organizes the adult brain to be more sensitive to triggers of aggression, excessive prenatal glucocorticoid exposure organizes the adult brain to be more sensitive to triggers of depression and anxiety. It's a different kind of programming, but the principle is similar. The prenatal environment, specifically the level of stress hormones, can permanently alter the brain's architecture and its vulnerability to mental health issues later in life. It's another layer of how early experiences, even before birth, shape long-term outcomes. To appreciate the next level of complexity, let's quickly recap genes. Genes contain the code for proteins, but they need to be activated to produce those proteins. Every cell has the same genes, but different cells activate different sets, determining their function. Epigenetics is the study of heritable changes in gene expression that don't involve changing the DNA sequence itself. Think of it as switches that turn genes on or off. Some hormonal organizational effects, like those from prenatal hormones, arise precisely from these epigenetic changes, permanently altering gene regulation in specific brain cells. A landmark 2004 study by Meany and colleagues in Nature Neuroscience beautifully demonstrated this. They showed that rat pups receiving more attentive care from their mothers, licking, grooming, nursing grew up to be less anxious, better learners, and had delayed brain aging. The paper revealed the mechanism. The mothering style altered an epigenetic mark on a gene crucial for the brain's stress response. Whoa! Mothering style alters gene regulation in the pup's brain. Even more astonishingly, these pups, as adults, became more attentive mothers themselves, passing this trait epigenetically to the next generation. Adult behavior produces persistent molecular brain changes in offspring, programming them to replicate that behavior. Following this discovery, a flood of research emerged, much of it from Meany's lab and collaborators. They found epigenetic effects linked to various hormonal responses, impacting genes related to growth factors like BDNF, the oxytocin and vasopressin systems crucial for social behavior, and estrogen sensitivity. These changes are pertinent to adult cognition, personality, emotionality, and psychiatric health. Childhood abuse, for example, causes hundreds of epigenetic changes in the human hippocampus. Stephen, Suomi, and Sif found that mothering style in monkeys affects over 1,000 frontocortical genes. The text calls this totally revolutionary, sort of. It highlights the profound impact of the environment on the very regulation of our genes in the brain. But, and this is a big but, enthusiasm for epigenetics needs to be curbed. Findings have been overinterpreted, and as the field exploded, the quality of some studies declined. There's a real temptation to conclude that epigenetics explains everything, whatever that might be, ignoring that most effects of childhood experience on adult outcomes probably don't involve epigenetics. Furthermore, many epigenetic changes might be transient rather than permanent. Criticism, particularly from molecular geneticists, has been sharp. Some resentment, perhaps, comes from having to incorporate seemingly mundane examples, like rat mothers licking their pups into their previously pristine world of gene regulation. It's a reminder that exciting science requires rigorous validation. Despite the complexities and potential overhype, the core message about epigenetics fits into a larger narrative. The real revolution here isn't just discovering yet another mechanism, whether it's synaptic plasticity, brain size changes, or epigenetic switches. The fundamental point, echoed by thinkers like Freud, Bowlby, Harlow, and Meany from vastly different perspectives, is that childhood matters. It profoundly shapes adult outcomes. All these fascinating biological details, growth factors, on-off switches, myelination rates, they're just providing deeper insights into the intricate steps linking early life experiences to later life trajectories. They help us understand the innards of that once revolutionary idea. Why does all this detailed biological understanding matter? Firstly, it provides concrete mechanisms showing how parents can influence their children's behavior, how adversity can create vulnerabilities, and potentially, how interventions might work. Secondly, and perhaps more tangibly, scientific validation can sometimes be necessary to convince policymakers. Think back to the example of demonstrating hippocampal volume loss in combat veterans with PTSD. It helped convince many that the disorder was real. Similarly, accumulating evidence from neuroscience and endocrinology about the lasting impacts of childhood experiences can bolster arguments for investing in good childhoods, ensuring health, safety, love, nurturance, and opportunity. Ideally, we wouldn't need such scientific proof to advocate for this, but sometimes the data helps make the case stronger. So, we've journeyed through the landscape of environmental influences, from the earliest moments in the womb to the complex tapestry of culture and peer interactions. We've seen how experience shapes the brain and behavior. But genes are also in the mix. Chapter 8 takes us back to the very beginning, to the moment of fertilization, when the egg and sperm unite to form the unique genome of that individual. How did the specific combination of genes inherited influence the behaviors we've discussed? Genes are clearly relevant. They code for neurotransmitters, hormones, receptors, growth factors. We are, in essence, an individualized orchestration of different gene versions. Let's explore how far we've come in trying to find the gene for that specific behavior. Discussing genetics and behavior carries two heavy burdens. First, there's the deep-seated suspicion, rooted in the dark history of pseudoscientific genetics, used to justify racism, sexism, eugenics, forced sterilizations, and horrific acts of violence. This history makes people understandably wary of linking genes too directly to behavior. On the flip side, there's the opposite problem: overwhelming enthusiasm. We live in the genomics era with promises of personalized medicine, people sequencing their genomes, and popular writing giddily calling it the Holy Grail. This often leads to reductionism, the idea that understanding the parts, genes, will automatically explain the whole behavior. This can foster essentialism, the belief that people have an immutable essence tied to their genes, reflected in studies showing biases based on blood ties or identical twins. This chapter aims to navigate between these extremes, concluding that while genes are important, their deterministic power is often vastly overstated. Let's start by reconsidering basic gene function. The central dogma says DNA codes for RNA, which codes for protein. Protein structure dictates function. Sounds simple, right? But here's the twist. Genes don't decide when they're transcribed into RNA. Before the coding sequence of a gene lies a promoter region, essentially the on switch. What turns this switch on? Transcription factors, proteins that bind to the promoter and recruit the machinery to make RNA. Other transcription factors act as off switches. Crucially, what regulates these transcription factors? The environment. This immediately undermines the concept of genetic determinism. Genes aren't the sole directors. They're part of a complex regulatory system heavily influenced by external and internal conditions. And this environmental regulation happens at multiple levels. Intracellularly, if a neuron is low on energy, specific transcription factors get activated, leading to the production of glucose transporters. Locally, if serotonin levels drop, the neuron senses it and increases serotonin receptor production. Systemically, testosterone traveling in the blood can bind receptors in muscle cells, triggering cascades that build muscle. And most poignantly, the external world matters. A mother smelling her newborn activates transcription factors leading to oxytocin release and milk letdown. The point is, genes are regulated by everything from the cell's internal state to the smell of a baby's tushy. Genes don't operate in isolation, they respond to context, implementing if-then logic. If condition X is met, then activate gene Y. The plot thickens when you consider the complexity of regulation. With multiple transcription factors, the number of possible gene expression profiles explodes exponentially. Just one gene gives one profile. Two genes, A and B, can give three profiles, both on, requiring three different TFs. Three genes yield 7 profiles, needing 7 TF. 4 genes, 15 profiles, 5 genes, 31 profiles. As genomes get bigger, the percentage of genes dedicated to coding for these regulatory transcription factors increases dramatically. It's a feedback loop. More genes need more regulators, and those regulators are also genes. This suggests evolution favors complexity through regulation over just adding more genes. Organisms with larger genomes invest proportionally more in controlling gene expression. This focus on regulation ties back to mutations. Mutations in promoter regions, the on switches, actually occur more frequently than mutations in the gene's coding sequence itself. Back in the 1970s, Alan Wilson and Mary Claire King correctly theorized that the evolution of regulatory sequences is perhaps more important than the evolution of the genes themselves. This is supported by the fact that a disproportionate share of genetic differences between humans and chimps lies in genes for transcription factors. Evolution, it seems, shapes how genes are controlled by the environment, perhaps even more than the gene-specific instructions. It's about the switches, not just the light bulbs. So, wrapping up this exploration of the environment's profound impact, from the prenatal period through childhood and into the broader cultural context, the overarching message is clear. While genes provide the essential blueprint and the raw materials for building a brain and body, the environment, encompassing everything from the nutrients a mother consumes, the stress she experiences, the quality of parenting, the influence of peers, the prevailing culture, and the subtle dance of epigenetic regulation, acts as the master architect, the sculptor, and the fine-tuner. It fundamentally shapes the individual, turning the potential of the genome into the reality of a person. The environment is paramount.